So, as it was mentioned, IRCERCA is an association trying to enhance the network of Italian researchers here in Belgium and also promote their research and also accommodate the newcomers are coming, of course, trying to make them comfortable in Belgium and in the, let's say, host country. And I think this series event is really, let's say, answering to these three main points of the association, trying to also to disseminate as much as possible the research and the research done in the let's say, host environment, host country, in this case, Belgium. So this time we will speak about new perspective for cancer treatment and the role of basic research and its interplay with pharmaceutical companies. And we will have, as usual, the talk split in two parts. So let me first introduce our first speaker, which is Dr. Professor Francesca Rapino. She graduated at the Institute for Experimental Cancer Research in Pediatrics in Germany, and then she joined Belgium for a postdoc, already in Liège, and then she stayed in Liège for quite a long time until she became, in 2020, assistant professor at the University of Liège as chercheur qualifié research professor at the GGR University of Liège. And of course, she's I say of course, but it's not so obvious. She's the recipient of a number of fellowship and awards, but that make her very qualified to treat this topic. And then I would like to give the floor to you and join me in welcoming Professor.
Okay. So now expectation very high. The good thing is that we're few, so I can shout and feel still that I have a microphone. So today, so thank you for the introduction. I don't know if I'm the most qualified to discuss about this, but today what we decided to do with Pierre, that actually we worked together is a very long time, 10 years, is to basically do a couple of talks where we try to put a bit on the table what are the assets of fundamental research. So why still we believe that fundamental research is very important for the treatment of diseases, and in our cases we do cancer. And also why, from the public recipients, this is basically where I will focus more my attention now, there is always this idea that, research is very slow. So we do a lot of research, there's a lot of money going in. If you come from literature part, you will say there is too much money going in. And still the outputs are not so evident to the general public, which personally I believe is not really the case, but it's true that it takes a really long time. And so a bit to dissect what are the advances that has been made in cancer research through fundamental research. So to basically assess the problem, this is always a thing that I show to my children, I have to say. When they ask me what I do, I always say that, the work of a researcher is basically to go into a crime scene and to try to look the different kind of evidence you have around and try to put a scheme together. And this sometimes gives you a kind of model that will simplify very complex disease, very complex scenarios. This has, of course, good things and bad things. Simplification is something that is needed to give models. It's also something that takes away the rest of the things that you have around, and therefore in diseases can lead to an hypersimplification and therefore sometimes failures of treatments based on that model. But in general, we could say that when we do research, we try to find unexpected links, unexpected models in what we're studying. And in our case is cancer. Cancer is a very, very complex disease. So it's always funny when in newspapers you hear, okay, you have cure for cancer. This basically doesn't mean absolutely anything, because cancer is one name for a multiple disease, a complex disease. Why? Because cancer can arise in every part of your body. So you can understand, I think, very simple, that breast cancer is very different from a prostate cancer or from an ovarian cancer. But also, and this has been years and years of excellent research, and I would say in the last 20 to 30 years, each cancer is actually composed from different types of cells. that of course proliferate, but they're also different one another. And so you have a different in cancers from where they start, but also a different inside each cancer, intratumoral heterogeneity that makes it extremely difficult to tackle this disease. You'll say, okay, this is already very complex. In reality, it's even more complex because what happens is that you have cancer and cancer gets treated now. And so you have cancer becomes a kind of chronic disease, thankfully, because it means the treatments work. But this means that cancer cells adapt continuously to your external stimuli. This can be treatment, it could be an hormonal change, you go in menopause, you know, we all do sometimes. And this change the kind of cells that compose your cancer, making it even more difficult to treat the cancer because the therapy you had day 1 will not be effective at day 3. Why? Because the cancer evolved, and therefore the mechanisms that you study at day 0 in your lab research are not any more involved at day 20 or 2 years. And this is why also research has adapted to understand this kind of adaptation and changes in the cancer biology, I would say. So this is where we are now. So what I showed you before is what we understood of cancer 10 years ago. And this is what we know of cancer today. As you can see, it's a much more complex cartoon. So cancer is, from the basic, a genetic event that happens in your cell. So your DNA gets mutated, your cell go crazy and start proliferate a lot. Of course, this proliferation also activates the surrounding, because if not, we will have cancer all the time. So generally what happens is that the immune system recognizes the cells that go crazy and kill it. And sometimes when you have cancer, the cell is able to escape from this immune modulation, let's say. Then the cells evolve, as I said, in time, and what they have as capability is to start moving. So they enter in the blood and they go in the other location. And this is what we call the metastasis or secondary location of the cancer. And when you treat the cancer, what happens is that those cells, they also adapt to therapy and they become resistant. So today, you can see, I think from the cartoon, that the complexity of studying a disease like cancer is extremely high. And this makes I would say interdisciplinary research, so people from different fields, from artificial intelligence, molecular scientists, clinicians, need to collaborate to really assess the dynamic of cancer in time. So why is cancer? So I think that then this slide it becomes really obvious. This complexity also reflects in high incidence and mortality of cancer. High incidence, I always go cautious there, is due to 2 reasons. One is because we are getting all older, which is a good thing. The second thing is because technology and therefore research has really advanced. So now we can detect cancer much more often. And so before, a lot of cancers were undetected. You were dying because you had a heart attack, but in reality it's because you had lymphoma, and we just didn't know. And so the high incidence of cancer is in reality also a reflection of increased diagnostic and technology. On the other side, of course, we still have a huge mortality. So there are a lot of cancer, breast cancer, lung cancer, prostatic cancer, they are still heavily killing. So the prognosis is still not so favorable, I would say. This reflects from an economic burden to society. So all over Europe, cancer is today one of the most expensive diseases to treat. One of the reasons is because there are a lot of patients. The other one is because treatments are very, very costly. Precision medicine, I will just talk about this very quickly, is something that is extremely interesting to do, but is extremely costly, and this increases massively the cost of the treatment per patient per year in Europe mainly. And of course today this economic burden is also something that it's reflected in the need to find new technology that makes cheaper the delivery of the scientific research into clinics. So this is a slide that I always show. I say, okay, if you do so much research, if there is so much advances, why we still don't have a treatment? And one of the things that you always hear, you know, when you go on Facebook, because yes, I still use Facebook, which I think is I'm the last one, is yes, the reason is because big pharma, they have the treatment, but they don't want to share it, due to the fact that, you know, then you will not have the disease anymore. If you see this graph over here, if this was the case, a person, a company that would develop an efficient treatment with cancer, it would be the most rich person all over the globe. So the fact is that the reasons why there is no treatment is not because there is no research or no development or hiding, is because the disease is extremely complex, and the hypersimplification that we have in communication makes, I believe, this is my personal opinion, general public think that there is something hidden just because it's hypersimplified. Because if you look at the numbers, everyone would like to find a treatment of cancer and sell it. So how do we work in cancer research? There are two, I would say, big worlds that generally discuss, and there is a big contrast over there. There is the business, so the companies. As you can see, there are a huge amount of companies. This is all over Europe, biotechs, pharmas that develop different kind of therapies against different kind of cancer. And then there is the university, so the Institute of Research, that actually are responsible for the fundamental research. Fundamental research is mainly, I would say, 99.9% financed by us, with our taxes, with our donations, with basically this, with our taxes and with our donations. And there are, in Belgium, very good financial, I would say, supporting agency. We are founded as a lab with that, that select, projects that can give rise to novelty and innovation. And of course, you also have all the international sponsors that help, the research. And one of the questions, it's always to say, okay, what is the difference between the companies and the research in the lab? Why the companies cannot do fundamental research and why the university cannot develop a drug? Because it looks pretty much the same. And in reality, the fact that you have two different entities, personally, this is my personal opinion again, I have to say this, I think is quite a resource, because the way you approach the problem when you develop a drug or when you look for a biological mechanism is completely different. What we do in university, we tackle the problem to understand how cancers develop, change, and adapt. Not because we want to develop drugs, of course, this is beautiful, it's nice that we go there, but because we want to understand the mechanism behind. We basically want to understand the mode of action of our specific protein, RNA, DNA of interest in that context. And so what we do is that we put for the public use the identification of new biological targets that have a specific mode of action that triggers a function in your cancer. What companies do is that they take their targets and they assess if they're druggable. So what they say is that, okay, this specific protein A, this specific target that the fantastic researcher in ULB found, can I actually produce a drug that can bind to this target, that is not toxic for the rest of the organism? And for that, you need a huge amount of time and resources and expertise that university today, I think correctly, do not develop. Because it's a complete different mind setting. There, what you do, you want to develop a product. What we do, we want to discover a mechanism. Of course, this doesn't mean that we're not linked, because if you see the graph over here, research, different kind of research, fundamental research, applied research, clinical research, have been the key propulsor of every kind of new technology that is today used in the clinic. CRISPR-Cas9 is one of the most famous one, because it has been the Nobel Prize really recently. This has been a pure fundamental process research that has been translated then in a product for gene therapy today. And we can say this about many things, COVID vaccine. This is all fundamental studies that have been developed from companies, by companies, to become a product. And so the talk between academia and business is actually extremely important to develop good drugs for the patients. So why does it take so long? It looks simple, you are in the lab, you do your stuff, find the target, you go to Pfizer, and Pfizer will develop your drug. And in 2 years, you should be in the market because they have billions. It would be beautiful, but this is not really the case. So fundamental research is extremely long. What we do, when we do research in the lab, we found our target, and then, so we found something that may have an effect in cancer cells, and then we start validating our hypothesis multiple times in multiple models that are all the time, as I told you at the beginning, are a very small reflection of what is in clinics, because cancer adapt. So you take a mouse, the mouse will have a cancer at day 0, but you will never be able to know in that mouse what is at day 20, because you stop at day 0. And so you have to do multiple rounds of validation. And thankfully also in the community, what you need to do is that if I discover something, I need also Pierre to confirm this discovery. If not, is not really accepted from the scientific community. Then you have to characterize this, and this takes, to have a good target, nearly between 10 to 15 years. To have from the scientific community the idea to say, okay, this target is valuable. It's something that really works in multiple models from multiple labs around the world. From that, you pass to industry. And this becomes extremely difficult. So if you have to do a product that is diagnostic, so basically when you go to take your blood and see if you have markers of cancer, to develop those kind of diagnostic kits, it takes around 5 years. To develop a drug in oncology, you go between 10 and 50 years. It costs around 100 million euros to go from the start of that drug discovery campaign up to the end with 95% probability of failure. So this makes not only very long, but also very costly your drug at the end of the day. And this is why it's still very long, the process between when I start in my lab to check if my cells die or not with my idea, to have a drug that can be given to my grandmother if she has cancer. This doesn't mean that there's not been not only an acceleration, but a huge change in cancer treatment, and so cancer patient prognosis during the year thanks to the research. So here's just, I would say, a kind of overview of which kind of treatment are today available thanks to the basic research that's been done all over the world. And down here, so the dates that I'm putting here below is because I would like to give you a very brief overview of melanoma, is what we are focusing on, and Pierre will give you a more in-depth, I would say, idea of what we do actually, we have done in the labs. But is basically I think melanoma is quite an emblematic cancer, showing how much the research that has been done on this kind of cancer has improved massively the prognosis of patients. So melanoma, very briefly, is a skin cancer. It's the most common skin cancer. It develops from melanocytes, which is why we become all brown and nice when there is sun, not here in Belgium, but in Italy. What happens is that in melanocyte, as in every other cell type, you can develop DNA mutations. Those DNA mutations are the trigger, so the starting point for malignant transformation, therefore pre-cancer, I would say, lesions, that then can grow into those very ugly, uncontrolled proliferating cells that are actually the cancer. Why melanoma is so deadly is because those cells are also very plastic, so they can move very fastly to the blood and spread all over the place. If you see the TAC, so this is a PET scan of a patient, all those dots, those are all melanomas. So he has basically melanoma all over the place. Those are all metastatic melanomas. So each dot is different from your primary tumor. So melanoma has been one of the cancers due to the fact that the models are very well established from a lab perspective. To be treated with all, I would say, the different developments of research and then directly drugs afterwards. Here I will just give you a couple of an example. The first one is chemotherapy. I use this because it's a very old treatment, still the most effective treatment that you have for general cancer. Why? Because it's based on a very general, very simplified mechanism. So to go back to what you told you before, when we look and we do research, we simplify. Sometimes this simplification, even though it's not reality, points out the key difference between a healthy cell and a cancer cell, and this is growth. Cancer cells, they grow a lot. And therefore, when they started to see that there was this difference in growth and start to understand that this was related to the fact that the cells divide, so the DNA is duplicated and then divided, they say, okay, this is kind of vulnerability for cancer cells compared to healthy cells. And so many companies developed agents, so systemic chemo, that is actually targeting the DNA replication machine, not because it doesn't go on your healthy cells, in fact you lose your hair that grow, but because it will tackle more, will kill more your cancer and less your healthy cells. And then localized radio that is based on the same idea, so you actually target more cells that duplicate their DNA more fast. Of course, even though chemo is very efficient in melanoma and many other cancers, has a lot of side effects. So when you go to hospital, people treated with those kind of therapy, they suffer very much, they vomit, it's a very hard therapy, radio a bit less. you have at one point a loss of response, so you develop resistance. So at one point your tumor, because it's developing, it adapts to that, to the drug, and you stop responding, and therefore you have a high rate of relapse. So what happened in melanoma, this was really, in the field of melanoma, very interesting, is that when those papers came out, so the basic research came out, you start to do sequencing of the DNA, was great, and so they start sequencing many, many cells. All over the place, all your DNA, you can know precisely what is the sequence, so what is the writing inside your healthy cells and your cancer cells. And so, of course, geneticists said, okay, let's merge them and see what's different. And melanoma was a very emblematic cancer because 50% of patients, they had just one single mutation in their DNA in a very specific protein, which is the BRAF protein. This BRAF protein, what it does, it boosts proliferation. If you have the BRAF V600E, that is the specific oncogenic mutation, so it drives cancer, your cells will start proliferating like crazy. This was actually golden because people thought, okay, maybe it's not for all the patients, but the patient who will have this mutation, if I find a drug for this protein, it will be very specific because normal cells, they don't have this mutation. So it will kill everything. And this is exactly so this was the first drug developed upon this research, so upon this identification, is vemurafenib. It's now not used anymore in this form, but it's the same principle. It targets specifically melanoma that has BRAF mutated, 50% of the patient. And here you see a scan of a patient, a real patient. All the yellow things, those are cancer. After vemurafenib treatment, no cancer anymore. Was beautiful, New England Journal. Everyone was excited. 6 months after, 100% of the patients that were treated with this drug, and they are still treated with this drug, have relapse and resistance. Why? Because, again, when you simplify a model and you look for something static, like is a mutation in the DNA, you don't understand, this is true, it happens, but you don't understand the adaptation. And so what happened here is exactly that the simplification gave a treatment that is not as effective as we thought because there is a development. And so you need to recomplexify again, which is what we do now. And so today, those treatments are still used, but we know that there are limitations. And then the final, I would say, development for melanoma is much more recent. So this has started to be first-line treatment in melanoma in 2020, I think, or 19. It's based on, again, a very important discovery that has won the Nobel Prize, I think, in 2017 or 18, where researchers for many, many years have started to assess that our immune system actually recognizes cancer cells. And cancer is developed only when your immune system is not able anymore to recognize your cancer. And so they start saying, okay, how this is possible? And they start really looking at the interaction between the immune system and the cancer cell, and they discover that cancer cells are smart, and what they do, they take out some proteins, so some proteins, some effectors that are actually blocking the immune system. Of course, this is not for free, because to produce this, they also produce a lot of rubbish, those tumoral antigens that are specific to cancer cells, but not to other cells. So again, this took a long, long time. It's still very much in development to understand really specifically this mechanism. But people started to say, okay, we could use this. We could use this to develop clinical applications. And so what they did, and this is now in clinic, and I have to say is a revolution in cancer patient treatment. So here, melanoma patients, some melanoma patients that respond to this therapy are cured. They develop some inhibitors, or something that blocks the thing that the cancer cells put out to say to your immune system, don't look at me. So you take away this, you take away the brake, and your immune system recognizes the cancer and kill it alone. But also, they activate, so they engineerize your immune system. This is the CAR T cells, so everything that is genetic modifying gene therapy. They modify your immune system, so they inject alive cells that specifically recognize this rubbish antigens that only the cancer cells have, but that your healthy cells do not have. Also here, good response, extremely costly. So this is extremely costly because it's really patient-derived. So the general idea here is that from a fundamental mechanism, that is to understand how the immune cells escape or respond or recognize a tumor, you actually develop today new generation treatment. There are, I have to say, one of the most amazing things you can see in clinics at the moment, even though, of course, you have people that do not respond with all the limitation of therapy. So what is the take-home message of today, after that I annoyed you for 30 minutes? It's basically to say that cancer is a chronic disease, and this disease is driven by cellular adaptation. So that cells, if someone is diagnosed of cancer in day 0, you already know that at day 365, 400 or 500, that cancer will be different from your first cancer. And this makes it very difficult to treat. And of course, it needs a continuous research on those cells, on elaborating new models, new concepts to find clinical options for patients. That fundamental research, I hope, by the example I show you, bring unexpected therapeutic solution. Because if you think outside of the box and you want to understand mode of action and mechanism, not because it's useful now, but it could be useful tomorrow, this brings you the liberty, I would say, that the university gives you to go a bit outside of what is directly targetable today. And this makes then innovation. And also, and most important, that innovation takes a lot of time. And therefore, that it's important also for the safety of the drugs that we bring to the market to give this time to every actor of the chain to make the good decisions, I would say.
Thank you very much, Professor Zuppino. So let's now go to the second part with our second guest, Professor Close. We started with a master in pharmaceutical science in 2003 in University of Liège, then getting his PhD later in 2006 in biomedical and pharmaceutical science, always at the Faculty of Medicine in Liège, and then staying as a postdoc, and finally getting in, it's a leadership position in 2013, ahead of the lab in cancer signaling. Is now a FNRS principal investigator and senior group leader always at the Faculty of Medicine of the University of Liège. So he has the whole path through to that university. He has been awarded several prizes. The last one, Worldwide Cancer Research Award in 2023. So it's very recent. Congratulations for that. And is a amazing curriculum in genetic mutation and all these. Let's say life science topic. So welcome and looking forward to your talk.
Thank you. So thanks for the invitation, and I guess the goal of today is to tackle a little bit the questions of what would be the impact of developing fundamental basic research for such a complex problem, such a complex disease such as cancer. So I'm not a pure product of Liège. I actually also spent a few years in London in an institute called Cancer Research UK. And why do I want to stress this out is because I had the question from my parents when I left to London. So I worked for an institute that was called Cancer Research UK, but I was actually working in yeast. So in a very simple model organism that has many advantages, but it's absolutely not a cancer cells. And many of the groups at Cancer Research UK were actually developing models based on yeast or C. ligands, or very basic fundamental models. And that's, I guess, where I learned the potential of developing really basic hypotheses from fundamental research. And of course, there you want to bring this to something that looks more like disease-relevant model. One of the things that maybe I think it's important to mention is, of course, I do basic research, we do basic research, but what we don't want to do is to oppose basic research to other kind of research. We have clinicians around us that actually look for patients, they study patient biopsies, they study patient samples, they raise conclusion. We do something else. We modelize the disease in a very simple organism or very simple environment. And my personal feeling here is that the two are actually extremely important and also bring different conclusions that one has to somehow assemble and make people work together to understand and to find new therapeutic solutions. So for example, if you are clinicians and you actually take biopsies and look at what is this cancer about, what are the characteristics of the cancer evolution, you will make observation, you will raise conclusion, but you will never be able to understand if what you see is causative to the cancer. So in other words, if it's driving cancer disease or if it's just a byproduct of the disease. Us, in fundamental research, we do the other way around. We go very much simplistic, but what we actually assess as a question is how much is it causative for the disease evolution. I think those are the two main advantages of the two research, and I really want people not to oppose those two research, because us, with our simplistic vision, we cannot do good research if we don't put this in patients. And clinicians, if they don't really assess new hypotheses that come from basic research, it's very unlikely that they will find an unexpected, innovative therapeutic solution. And in that interest, so we work at GIGA Institute at the University of Liège, and this was an initiative many years ago when I was a young student to put all the people involved in biomedical research into the same place, which is the institute. And this was very important for the management at the time of the university to say we want the biomedical research to be in the hospital. We want the basic research to be next to the patient, next to the clinician, to really force and highlight potential collaboration between the two main drivers of biomedical innovation. And this is where we are now, and GIGA is an interesting place because we have people, it's not a cancer research institute, we have people studying developmental biology, we have people studying chronic inflammation disease, and we have a cancer research department. It's fair to say that cancer is a disease, but it's basically a caricature of an hyper-developmental process that basically is uncontrolled. So if you understand developmental disease, you can also make hypotheses that are relevant for cancer research. So that's the idea of putting people from many different disciplines in the same institute using different models, but at the same time trying to provide solutions for patients. So as Francesca said, we are sponsored here by many different public or private foundations. And I have to say we are quite lucky because basic research is costly. It also takes time, so we have the luxury here in Belgium to have people around us understanding that this takes time, that we are allowed to do mistakes, we have the right to be wrong, and with this required a lot of education and also understanding. Of course, it's actually linked to excellence. I'm not saying that we should not promote excellence. So if we go a little bit more on what kind of hypothesis we have developed in the lab over the last 10 years, it starts a little bit with what Francesca was showing earlier. So there is this observation. It's quite obvious. If you speak to any clinician, they would tell you this is extremely complex. We don't really understand. And if you have patient A, patient B, it looks, we have some similarities, but we have also a lot of differences. And the heterogeneity means that you have many different things that sometimes are similar, but very often are different. And what Francesca highlighted here is very important, is that not only two patients are different in terms of cancer, even if you have two women with a breast cancer, they can be of different features. So this is what we call here inter-tumor heterogeneity or inter-patient heterogeneity. So you will not treat patient A and patient B the same way because they are different, even though they look the same. But what is even more complicated for clinicians is the fact that within the same tumor, there seem to be different subtumors. So in other words, if you would give a treatment that would trigger the red cells here, the blue cells will actually be still going on. And so there is this heterogeneity, this diversity in the cancer cells that makes the problem extremely complicated. And Francesca highlighted the fact that this is a chronic disease, so it means that those three, let's say, subpopulations that are designated here, they would evolve differently over time. So if you treat the cancer here with a red treatment, you kill the red cells, you will see that the green and the blue are going to evolve differently. And this is something that was quite striking with the sequencing techniques that are all over the place. But this, as you can understand, makes the problem extremely complex for molecular component. And this is basically at the core of treatment failure. So conclusion, which is quite pessimistic, right? So if you speak to someone that has the disease, you say, well, how am I going to be treated? And me, as a cancer researcher, it's very difficult to accept the fact that whatever we do, we cannot really find a good solution that is realistic at the same time. Because there is the idea of studying the cancer for, intellectual, let's say, evolution. But there's also us as cancer researchers, we want to find solutions or, let's say, solutions, treatments. So the heterogeneity is really what causes therapeutic failure. So this is why people get somehow cancer evolution. But what we started to develop as an hypothesis is that despite the fact that cancer cells are going all over the place, they change over time, still what we see is that there are common mechanisms among the blue, the green and the red that seem to be important for the acquisition of a function of the cancer cell. So a cancer cell has to be proliferating a lot, has to be migrating because it's going all over the place, and it's going to be resistant to the cancer treatment. So despite the fact that they look different, they seem to use mechanisms that are quite similar, but it's not written in the gene. So that's what I call a conversion adaptation mechanism. And those mechanisms are actually coming from, and this is where I believe basic research is important, they're coming from the intrinsic machineries that is available in the cells. So they basically use developmental processes. So the processes that have been in place during development that are silent, and that are basically highlighted during cancer resistance or cancer evolution. And so what we speculate is that despite that there is this very massive heterogeneity, we can still, or let's say, we speculate that despite this heterogeneity, we can still find simple mechanisms that may be common to these different cancer cells. And we think that this is by co-opting or reprogramming processes that have been set up at the developmental stage in the cells. And those fundamental processes, they become somehow extremely limited for cancer growth, metastasis, resistance. So we believe that they really become what drives some specific cancer features. One of the basic mechanisms of machinery that is present in all the cells is the capability of cells to synthesize protein. I mean, you start from the DNA, and then you synthesize protein, which are the, let's say, the workers, the effectors in the cells that they make, that's what give the function of the cells. And this is basically what we study because we believe that the way the cancer cells are going to synthesize protein is different from the normal cells, and it's quite common between the different subtypes. And so we asked the question, can we somehow study this specific process of, production of proteins to find new therapeutic intervention possibilities. So what we want to see here is to highlight the vulnerability of a cancer cells towards an healthy cells. So would a cancer cell be more susceptible to this particular process of protein synthesis versus an healthy cells? And so this was, let's say, the starting hypothesis of the lab. And it's based on a simple observation is if you compare, this is quite simplistic, But if you compare normal cells to a cancer cells, you can see that, so the structure here is a protein. We actually see that protein synthesis, the production of protein, is different between a normal and a cancer cells. And it's different. It's not only that you produce more protein, you produce protein in a different manner. And we believe that this is very important to highlight this, red kind of protein that you see here. And those proteins, we believe, are the proteins that are driving specific cancer function, such as the survival, because the cancer cells hyper-survive, the resistant invasion, and also metastasis formation. And so we ask the question, quite simple, what is it that you need to go from this situation to this situation? So what is it that the cells has to put in place? What kind of tools the cancer cell needs to produce this? It's not that you want to interfere with this, you want to prevent the synthesis of the red proteins. And what we basically discovered over the years is that in the process of protein synthesis, there are small molecules, RNA molecules, that are very important for the transition between these normal cells to the cancer cells. And those molecules are RNA molecules, they're called transfer RNA. And you know, in biology, if you had a bit of biology in your background, transfer RNA are the key molecule that read the genetic code and translate this into a protein. And in this world of those small RNA, we can see that there is very much specificity between normal cells and cancer cells. So what we started to do in the lab, and I will give you a couple of examples, is to say, okay, if those molecules are so important for the establishment of a cancer cell, so for the cancer cell to be functional, to be doing what it does, it survives, it migrates, it resists, then can we generate, engineer models to specifically remove those molecules and see whether you actually would impact cancer development. And so we started to assess the possibility that those small RNA molecules would be new therapeutic strategies or new therapeutic options. So what we started to do is to say, okay, what are the red proteins here that seem to be very important and distinguish the cancer cell to the normal cells, whatever they are, and to say, can we define what they are and how important they are for tumor growth, for the metastasis formation and therapeutic resistance. And of course, what we want to do is to interfere with the production to see how much we can prevent cancer features, cancer phenotypes that we say. And this is, let's say, the first publication that we did in the lab, the first evidence that, our hypothesis was making a bit of sense. As I said, when we want to study a molecule or a function in cancer biology, we build models. And here in this case, those are mouse models, so those are mice. And what those mice do, they actually will spontaneously develop, in this case, colon cancer. So over time, the mice, it's a model, so it's very well established. It will spontaneously have intestinal cancer. And you can see that this is an intestinal villosity, epithelium, tissue. And over time, you will see that the mice is going to develop small tumors in the intestine. And this is a model that does this. What we do is we use that model, and genetically we change the model, and we remove some of the molecules that we believe are important in this process. And so we always compare, let's say, a colon cancer mice with a colon cancer mice that doesn't have the small RNA molecule in place. And what you see here is that if you, so I will go straight to the final data here. So the colon cancer mice, you can see that over time it will develop, this is an intestine, a lot of different small tumors. You can actually see them, here differently. And when we actually started to look into those small RNA molecules, we could actually completely, it's not a small effect here, completely prevent tumor onset, tumor formation. So suggesting that here we have a small molecule that seems to be essential for the process of tumor initiation. Of course, this is a genetic model, so I mean, here there is still a long road if you want to think about a cancer treatment. Importantly, because when we speak about cancer treatment, we always look at what is a normal organism responding to this inhibition, and here we have mice that do not have cancer, so they will never develop cancer, and we actually do the same thing. We prevent this molecule to be produced. And what we see is that basically the normal mice, so that has no cancer, a healthy person, like hopefully or today many of us, have no consequence if we remove that molecule. So we seem to have something that does not really perturb the normal intestine, but if you have tumor onset, then you actually here completely inhibit tumor initiation. We have done at the same time, basically, because we use different models. This is another model. It's also a mouse model of breast cancer. This model is quite particular because not only will develop, this is a mammary gland in mice, so you will see all the branching here of the mammary gland, so the organ that produces milk. And what we do is we have models of mice that spontaneously will develop breast cancer over time. And this particular model that we used is developing metastatic breast cancer. We know that after 12 weeks old, those mice will develop metastasis in the lung. And again, we genetically inhibit our small molecule here that is a small RNA. And what we see is that we actually not only prevent tumor development, but we also very strongly inhibit metastasis formation. So this is fundamental research. So we can say from that point that it looks like here we have identified a key small molecule here that is very important for the development of colon cancer or breast cancer. This was very important for the scientific community, but it doesn't really tell us still how we can use this to think about new therapeutic avenue. So we started to address that question. What is the clinical potential of, so can we highlight new therapeutic solutions? Is it clinically important to inhibit those small molecular RNA? And we tested this hypothesis, and this is basically the work of Francesca when she started in the lab in 2014 to assess the clinical potential of inhibiting those small RNA molecules, and specifically in the model of melanoma. So I will quickly speak or re-say a couple of things about melanoma. Why did we go for melanoma? For many of the reasons that Francesca already explained. Melanoma is it's a terrible cancer, so it's not very frequent, but it's extremely aggressive. It's the most aggressive skin cancer, and it's extremely resistant. So it's really reluctant to any kind of therapies or back in the years. And the reason is that melanoma arises from the skin, of course, but quickly it will actually penetrate into your dermis and reach the blood circulation. And melanoma is very much malignant, so very quickly you have metastasis. And most of the time when patients are actually entering clinics, they're already at the stage of metastatic melanoma, which is linked to the bad survival of the patients. But as Francesca highlighted here, all the sequencing of efforts that were done on melanoma patients highlighted half of the patients that have this specific BRAF mutation. And this is quite unique in cancer biology, that you have such a conserved mutation that is responsible for the disease. And this was the opportunity for people to develop this one of the first target therapy that would specifically target this BRAF gene. Why am I highlighting this very much? Because it's been, so if you think about basic research, this is for us quite an ideal model to test, therapy resistance in a system that is quite simple, because it's from a genetic point of view, here you have half of the patients that have the same mutation. So you remove complexity a little bit. As was mentioned from Francesca earlier, This was a massive advance. This was in 2011. This targeted therapy, I remember I was a postdoc at the time, and I was like, okay, so we have found the solution against melanoma. This is done. This is very efficient. But unfortunately, it was highlighted that most, if not all patients, eventually, even though they really respond very well to the therapy, they at some point become resistant and they die from melanoma. Why are we interested in melanoma here? Is because it's quite a model cancer for clinical research. We have access to clinical data, and this was very important for us. What we basically did here, we went to databases and we looked at data from patients that are melanoma, responder to the therapy or resistant to the therapy. So over time they become resistant. So here, what you see is that we can group the patients that are resistant, extract the data, and predict from those data what are the red proteins that are somehow driving the resistant phenotype. So what is it that you need to produce to be in this red cluster and to basically respond? And of course, when we looked at the resistant patient, and this is why we started this project, we found that those specific tRNA, transfer RNA, were particularly important in the resistance subtype of patients. So without going too much into the data, this is for us a very important experiment that was done here. So of course there's a whole publication here. This was published in Nature by Francesca in 2018. This has been quite a big discovery. But what we did is we inhibit genetically our small RNA mechanism here in patients that are either sensitive or resistant to the therapy. And you can see here that if you compare the black line with the red line here, you can see that the patient biopsies is resistant. So if you treat with the therapy, you don't see any effect on the tumor. The tumor is not responding, so it's basically growing under treatment. But if you genetically inhibit our tRNA mechanism here, you see that you really specifically inhibit the tumor under treatment. So you basically are able to sensitize a resistant cells to the therapy. This is tumors that have been implanted in mice. So you treat the mice. So what we see here is that we not only we have something that seems to be important for cancer biology, cancer development, but we have something that has a clinical potential. It means, in other words, that if you find a way to pharmacologically prevent this small molecule to exist, you will be able to combine this with the existing therapy to prevent resistance. So in other words, during resistance, you have this specific small RNA that is popping up, that is driving the resistant phenotype. If you prevent it, you resensitize the therapy. So in other words, what we believe we've done over the last years is to really highlight a new mechanism, new molecules that are driving the resistant phenotype in malignant melanoma. And that those mechanisms could actually be somehow at the origin of new therapeutic solution. In melanoma, of course, but over the time now in the lab, what we study is the potential of this mechanism in multiple resistant phenotype in cancer. Of course, here we're talking about combination therapy. I think this is something we didn't really talk about very much, but the future of cancer therapy is the combination of different treatments. Because if you acknowledge the fact that cancer is an heterogeneous disease, it's impossible to treat this disease with one single treatment. So you probably need, at the same time as using a target therapy, the BRAF inhibitor, you can also add to that treatment the drug that would prevent the resistance to this therapy. And so, let's say the conclusion is that we have highlighted here a new vulnerability in cancer, so a new weak point, a point of action, and that may highlight opportunities for the development of new therapies. So what do we do with this? Well, we're happy, of course, in the scientific community, this was the first time that those tRNA were somehow highlighted as important for cancer. But us as cancer researchers, we don't want to stop here, of course. We want to see how much we can drug this. Can we develop treatments that would mimic our genetic models? And this is something we have started to do, also together with Francesca. We have started a small biotech company, so this is outside of our academic work. So we basically raised private money to be able to design drugs against this small molecule here. This is only the beginning, but the idea is to create sufficient industrial knowledge to be able to pass that to a pharma company in the next 3 to 5 years. Of course, here, we want to study more, tRNA in cancer to do that. I mean, I always like to show this because it looks simple. It looks that we do models, but this is the work of many people. Here, Francesca is not in this picture because she left the team. This is the actual team now. So this is the actual team now. I mean, PhD students, postdocs, technicians, but also it's a highly collaborative effort. We collaborate with people here in Brussels or in the UK, in France, in the Netherlands, a bit all over the world, to make this happen because we believe that collective effort is needed to bring new knowledge and new innovations. That's what I wanted to say today.
So thank you very much for this presentation. Very interesting. So I would like to invite you over here. I think that I would like to start with a very general one. So it was mentioned in your presentation the time that is needed for reaching the treatment from fundamental research. And I would say that in the news, what we see, like I make an example, sequencing of genes, it happened in '72, but now we can do it in a matter of nothing, and that's
$500.
yes, just a matter of cost, and we see that this progress is like evolving at a fast pace. And is this the same for the time that is needed from going from the fundamental research to application? Is it decreasing, or is still somehow fixed and blocked because I imagine regulations or complex problems? So how do you see this? Maybe start with you if you.
So it still takes time. So why is it taking so much time? I think we highlighted today the time of research, but we also need to acknowledge a fundamental question is that because in the news, people doing shortcuts. What we do is we highlight possibilities. We're not saying that what we have found is a new treatment or a new, and this is sometimes what the news somehow the journalists want to put forward, and also sometimes scientists that want to oversell this a little bit. We have to acknowledge the fact that we still use models. So when you use mice, this is a simple genetic model, so it has one mutation. The patient, of course, the cancer starts with one mutation, but when the disease is diagnosed, you have a collection of mutations, so the disease is much more complex. So it's a bit naive to believe that what we found in simple models is directly applicable to a complex disease. And this, I have to say, we are a bit all responsible for this, because funding agencies, they have to show to the donors that there had been progress, and we sometimes want to go a little too fast in claiming that we have found a solution. I've seen colleagues or universities saying, okay, we know how to treat metastatic cancer. This is, to me, intellectually wrong, and if you may have a therapeutic solution, we live this in the context of the biotech. This may be true for one particular cancer or one particular subset of patients, but completely wrong for another subset of patients. And that, only time can tell. You develop artificial intelligence program, prediction strategies, methodologies. Still, you need to test this. So you would never take a treatment that has not been tested before. Unless you're really in a very advanced disease that basically you have no other options. But this is one of the reasons why it takes time still. And one of the challenges that the pharma companies are facing is that we have, and I think it's quite a failure of research, we have to admit this. We provide many solutions, but one has to test this on patients. But one of the funny, it's not very funny, but it's crazy observation is that there's not enough patients to test all the possibilities, right? So there is a competition towards having the patients to test your therapeutic solution. So so farmers, they say, okay, well, we want this subset of patients, but the other pharma company wants the same subset, and they actually compete to test the therapeutic solution on patients. So that it's a bit crazy to say that, but it's the reality. So there is, it takes time. We need to test those things. And you cannot just claim that you have the solution just because you did a couple of models.
Thank you. And maybe you have something to add to this. Do we have hope to reduce this time?
I think in general we reduce the time. It's just that because technology, so it's true, all this technology, you can see it also from the Nobel Prize, that comes out. Now the impact of technology on possible treatments is much more easy to detect. This, I think, it's the main difference between 20 years ago and today. So genetic sequencing, now we know, so you sequence, if you did the technology, it's clear that it will have an impact. This doesn't mean it's a therapy. And I think that this is a bit the difference. So you speed up technology a lot. The fact is that still, when you have to put them in patient, you have so much health, ethical problems. So people say, I remember read an article that said that the most advances in medicine were done during the Nazi period because there were no ethical law to treat patients, so to treat human beings, because they was doing in the camps. And if you think it's ridiculous, there were 4 Nobel Prize for medicine that came out from those experimentation, which of course I'm not saying, but what I mean is that there is also there is a balance between progress of what we can do, or the targets that we can do, and also the ethical boundaries of what is correct and safe, and where is that we can.
So for example, one of the things that is mostly developed now, before entering the clinics for drug discovery, is what people call organoids. So those are 3D tumors from patients that is basically maintained in a 3D kind of biologically relevant system. And what we can do with those organoids is you could pre-test your treatment or your combination to actually predict what you will actually test really on the future patients. Those are things that are being set up, put in place by universities, but also by companies that are expected to reduce the time, or to, let's say, increase the safety or the efficiency of the treatment.
I think also simulation will be very likely. So one of the things that is happening now is the engineering, so are really working on simulating environment. You remember when we went into space, they were doing a fake cell. So it's all in the computer. They simulate the cells by all the knowledge that they know of the things, and they make treatments. Everything, it's in silico.
Computation.
And then it's computation, and then you have to test much less combination of drugs to go there. So I think that I believe that this will be the greatest revolution to make it shorter, because there you will have to test less, so you will need less patients, less animals, less ethical issues. So I suppose that there is chance, and you never know, but I think that there it's so not from what we do. I think what we do, the time is limited by the models. I think we have to change the models there, and they're very likely, I believe, it will not come from biologists.
What is still difficult to predict from going from an hypothesis to a treatment is your drug could work on cancer cells, but it's not because it works on cancer cells that if you treat a patient, the cancer is going to be treated, because the drug has to survive the organism, be metabolized in a correct way, and this, taking a drug as a patient is not something that is trivial. It is quite challenging for the drug to actually be reaching the tumor and doing what it's supposed to do without having a side effect. And this is still very difficult to predict. We have animals to do that. Well, now we have, you know, a bit more ex vivo kind of assays, but it's still, you know, what is driving most of the failure in discovery.
The delivery, okay. But that's, yeah, good. Thank you for this
You have to stop us
answer.
now. We can go around for hours.
It's
You
very
know? We do this
interesting.
all day.
Well, okay, so I have another one if there is no question from the audience, and it's pretty much related also to some of your slides. Is what we saw, for instance, one of the pointing cases, the mRNA vaccine that was basically the framework was not developed for that, was developed for something else, and what do you see now, and what you're doing now, it's also applicable to other illnesses, and in general, like the fact that we are allowed, we can study more, say, platform to cure and investigate will help us in treating other diseases as well at the same time. How do you see this? Maybe this time we can start with Professor Rapino.
Gosh, it makes me feel so old. Also not. But I think that this is the beauty of fundamental research. I think is what, when I started as a postdoc in Pierre's lab, this was very fascinating for me because I came from a PhD lab where we were really focused on drugs in cancer. So we just did this. The idea that you can test, you know, mechanisms only in cancer. Instead, when you go a bit back on really fundamental mechanisms, you realize that, you know, you use a model. This model gives you an idea, which is a method in reality, is a method that a cell used to develop a specific adaptation to a phenotype, and that if you're lucky enough, you can tackle part of this mechanism, you can start to understand part of this mechanism. And then it's very, I mean, it's a speculation that you go directly after. You start saying, okay, if I found a method, so I found a structure, I can then use this method and structure to go on many other different models that adaptation is the base of life, is what we do every day. You adapt to traffic, you adapt to your husband, you adapt every time, and you normally as human beings, we use always the same mechanism. And so I believe that the RNA vaccine is actually a quite obvious now example, because RNAs are studying from, I don't know, ages. And the idea to then, because we know so much the fundamental biology, then we can use it as a treatment. And the idea is just, you know, I know, because I know RNAs, but I know also microRNAs, siRNAs, how they silence. And then there, because you have all this knowledge that looked very boring, then you have a treatment in a year and a half. A lot of millions of euros and dollars inside, but still a treatment in a year and a half. So I think that this is something that we should highlight much more, that what we discover is basically methods. So yeah.
And the other way around, what we see as cancer researcher is that the COVID, I mean, it's a bit bad to say this, but was, from a scientific point of view, a huge opportunity to develop something that was, you know, somehow, you know, in the pipeline for cancer research for a long time. But COVID actually, you know, was an opportunity to test this in a very large population, with some you know, emergency. But then what I see here is that now the mRNA vaccines are becoming one of the first therapeutic, you know, hope for the future of cancer research, which was not the case before the COVID. So you know, it's the other way around also, that, you know, an opportunity can also feed back to other fields like cancer research. I mean, that, to me, this is quite fantastic to see that now most of the companies involved in COVID mRNA vaccines are actually developing the same kind of strategy for other disease. Cancer is obviously a very common disease, but you can think about, you know, diabetes or different kind of chronic disease.
And also in this case, I believe for COVID there was no, let's say, shortage of funds and patient to treat or willing to treat, so there's no competition between the companies. And
also, I mean, I've never seen so many patients being tested for a solution in such a short period of time, and that never happens in you know, in the whole drug development process that I have experienced over the years. This has been a very impressive. I mean, we can discuss, you know, if it was good or no, but at the end of the day,
Me disagree there. We cannot discuss. It was good. I will state this publicly. There is nothing to discuss there.
but from a scientific point of view, this has been a massive opportunity.
And also this is, I think, with the COVID and mRNA vaccine, I think that this is highlighting another point. I think that you're also part of somehow, because this is fundamental research that was gathered and then was packed within a company, and then was licensed to another even bigger company. So there is a commercial mechanism that is always very delicate when it's medicine and the pharmaceutical environment. And so a typical question is like, why is research developed fundamental research is developed with public money, with public grants, but then the actual treatment is developed in an industrial environment. Is it needed? Is it the way it should be? Can it be another way? Or how do you see this? And I think you have you've seen both sides of the story, so you can give a thorough explanation or point of view. So first, it's
because if
difficult.
not, it will be too divisive. So go.
I think it's the way it should be. We can say it's quite abnormal that patients have to pay themselves for their own research, because foundations is basically funded by donors that are somehow close to the disease. Pharma companies, they do research, but it's also a big risk. I'm not going to defend pharma companies here, but it's still a big effort to do this. So there's a lot of risk. I mean, we can see this. We started a little bit the efforts here, and we can see that why did we start to be wanting to translate this into a biotech company as a lab? So you can ask the question. You have good funding in the lab, so why do you need to create a company to do this? I think that's the way to answer that. The reason why we started this company is because it gives you another level of ambition that you cannot reach in an academic environment. The questions that are raised by pharma, investors, market are completely different from the one that we are subjected to in the university. And it's different. It doesn't mean that it's better or bad. It's just different. I think they all have to coexist. The question that we have at university is how innovative it is, how validated this is. The question in pharma companies is how are you going to treat your patient with your solution? So that's, in university you have the freedom to claim this, but in pharma companies they will tell you, I'm not going to give you a single euro if you don't prove me that your solution is going to work. So there is much more pressure because, of course, it costs much more money, but also much more ambition. If your hypothesis work, if your tool work, then you can really give your knowledge, quite an important dimension to develop treatments, biomarkers, and have really the potential to be able to say one day, well, what we found many years ago, it seems to be quite credible as a tool to treat patients. It would be a mistake to believe that university can do that. I'm against that because I think university has still to innovate, to shake a little bit, what is known, to take the textbook and put them the other way around. That's what we have to do in the university. Pharma, they don't do that, and they take what you do and then they test this, and then they try to develop a tool that would be something that you would take as a drug. I mean, if I would develop a medicine in the university, honestly, you would not take it, huh? Because and it's normal. I mean, that's not the job of the university. I think I'm actually quite I'm fine with this, and I think. Objectives are different.
The question is also the return. I think the fact is also that what people always complain about is that it is true that the return of a success story in pharma is 95% economic return on this was the case of COVID. There was a big case. The technology was developed in Oxford and then was licensed in Moderna, and Moderna got all the return because the was without royalties, and so therefore, the return was very minor compared to the business that they made through the COVID. And I think I think there is a bit like the same attitude that we have when we look at success story. Normally, when you have a success story, you publish a good paper, you have a good
Survivor bias.
is exactly, so you don't see how much there was a risk for you to die in between. So, for example, for me, and this is to go in line with you, the university public funds, they cannot take the risk that a company does, because in reality, you have much more chance to fail than to succeed. And me, I would not be happy to pay my taxes to put in a thing that will be very likely unsuccessful. And of course, like everything, if you have this big chance of not succeed or fail, then you have to have a big return. If not, you will be completely crazy. In reality, is what we do also in academia. So when we go and we aim to go for big papers, it's not easy. Most of the time, it's not even funny, to be honest, having done this with you. And then, of course, you have a return. And then you say, okay, but because you succeed, then we tried many other times, we didn't. I mean, it's not, then you say the beautiful story, say, I went to nature, or yeah, I developed a drug, but in between, you were not happy, your hair is all white, yes, this is not natural, and there is a lot of, and so of course you need to have the return. And I think that this is something that is not very clear. There is a bit of taboo of saying this in the public. So university, we have the arrogance as university to think that we did already 95% of the job. It's not true, we did 2%, because we don't take the risk. Because me, if my publication is crappy, I still will get my salary. If I am the CEO of a biotech, I'm home. So I think this should be, this I learned I think we learn it by talking to those business people. There is a risk that we underestimate when we are in public.
But to bring a molecule that we're doing now to the market and to be able to treat patients, it's a billion of investment with 10% risk of success. So, I mean, I'm not saying that it's not like admirative. It's not that. But there is a massive investment and massive risk, and there, it's a reality. We have another reality in university. That's 2 different worlds. But what I like still, as a founder of the company, I like to be somehow still driving the science in the company. It's a different kind of science. It's different. I mean, you don't discover, but you develop things in a way that it's still, making sense from where you come from. And I think there's something very enjoyable there, that as a scientist, you have a simple model, and then now you're testing this on a collection of patient biopsies or different mouse models, and there is something enjoyable there, and that is very nice, because you see that the ambition is directly different.
Okay, so a solution that stays in the lab is not really a solution for the benefit of the people.
I don't think so. No. That would be a big mistake, and that would be somehow we should be forced to do this more. Honestly. Oh yeah.
I agree with you. We have a question from the back of the audience, please.
So I don't know if I can add something to this, but I actually used to work for Pfizer for the COVID vaccine. I won't work anymore there, so we can openly discuss this. And I think what is also important to know in that case is also to see the space we need to produce the vaccine. So I've been to a production zone, and some of the rooms are bigger than this one, because we need very big and huge equipment. And what is also very important to know is the amount of people needed to work in this. We have operators, scientists, we have a lot of different people, and that's why universities and pharmaceutical companies have to work combined, and of course each one of them have different values to bring, but I think that's also something that it's, I used to work in academic, now in pharmaceutical company, there was a very big difference, and the fact to see those very big production zones is something everyone, in my opinion, has to see.
Yeah, when we went, where were we? That we went to see those production zones for viruses was quite impressive. There, when you see it, I agree with you. You think, okay, the people, when you think you can do this in academia, where we are, you know, where your pipette's there and you say, oh yeah, it's so it's completely, it's unrealistic.
What we have experienced in the process of, you know, switching our discoveries towards biotech, that we of course discussed a lot with universities, tech transfer officers, and with funds, you know, investors, and what you can see is that what I really experience is when you talk to university, everyone is a bit arrogant in this world. So university, university, they you know they think that they know and you know that they can do it, and business they will tell you, no, you're a professor, there is no way we're going to invest on you to do a company. So we take your idea and then we need to have someone that actually is going to do this for you. I think, to me, this was an unreasonable arrogance between the two sides. Everyone is wrong in there. I think there is no reason why you cannot take your discovery and go to a company. And there is nothing wrong about this, because some colleagues at the university would say, yeah, but you switch a little bit. So now you are, you know, in this world.
On the dark side.
Yeah, on this world. I mean, to me, I'm always
We say
amazed.
this about editors too. We say when you switch to the editor side, we say that those are.
But this is wrong. I mean,
yes,
we should,
I agree,
you know, bridge those two worlds, because this is where, you know, the solutions are.
And I don't know if there is some other comments from the audience. And you have one? Yeah, please.
I think that we are all, je parle en français, c'est beaucoup plus fluent. On est tout d'accord que la révolution, c'est le séquençage. On doit faire le séquençage des tumeurs. Et une autre tool très puissante, peut-être la bioinformatique. In silico and sequençage and in silico analysis, I think that is a tools very, very, very strong.
Digital health is actually really exploding now.
Yeah.
Me, I'm a lover of in silico and artificial intelligence. I have to say, I've a bit switched from what I was doing before. I think it's amazing, but when you start looking at it, you see that there is, again, always the same thing. There is a complexity of those kind of systems. And I think that there, a bit to go back, it's very important to collaborate and to work with people who can validate your prediction. Because the beauty of artificial intelligence or prediction, you will always predict something. Yeah. If you do it in a silico screen for drugs, everything will bind to your target.
Also for vaccine.
Yes. You're very satisfied. You say, it's fantastic. You know, I found, 20,000 molecules that will work. And I think for training those models, the strong collaboration with the wet lab, which still is, I mean, a strong proof of concept of your prediction, is essential. And what I find now that I have a couple of bioinformaticians in my lab, it's very difficult for me to communicate with them, but also for them to understand that the math does not always reflect reality.
Is an informatic or bioinformatic?
Informatic.
This is the problem.
Exactly. No, she's very smart. She's a PhD student. But I see that, you know, you also have an advantage because you actually, they look at the problem in a completely different way. And I think that is also, I think, the same discourse, the same speech that is about academic and business is very naive to think that I believe in a work that is so complex, you are the only keeper of truth. Yes. I found this very fascinating.
The same experience I have. The same experience I have in Catania. I am a biologist first, and chemist of protein after, because in Sicily they have no work. We study. And when I arrived in the department of chemists, the chemist thinks that it's possible to reproduce a cell in a baker. No, it's not the same. It's not the same. And it's the same problem now with the informatic that work in bioinformatics. We have a difficulty to communicate, to large view.
Yeah,
okay?
but I think that this is, I think it's very fascinating, for example, that you have a person wake up in the morning and think that they can reproduce something in a becker. I think it's beautiful because they push boundaries, you know, and then you have someone else that goes and says, no, really, it's not like this, but this is still knowledge, and informatics is the same thing. It's just that I think you have to be humble enough to know that you're representing a part of reality, and that's the beauty. If not, we will be all out of jobs. I mean, you know, you do your thing and then it's done, and then your life is, you're 40. What do you do after? You know, it's already tough to be 40.
It's mandatory to use the same strategy of tumor, signaling.
You can say, you know,
signaling
there is
with
signaling
other
everywhere now.
Yes.
Okay, so I think that, yeah, we are close to the end. Maybe I have one last point that is always very interesting to me, and is because we saw it as a very complex tumor research is very complex because the disease is very complex. It's a iterative process, but this is true for science and technology in general. And what we see nowadays is that with all this media exposure, we have to go for dissemination as we're doing today, but with a bit of time. We have to go through some simplification, and sometimes just to have a headline, we go through oversimplification. So I have my own personal experience in my field, but like in your field, like how much this media oversimplification basically put a hurdle on, make your life science research more difficult. What is your opinion on that?
I think it's very bad, so I think that because what it makes you, it puts you in the position to give very strong opinion, you know, because if you ask me a question and I have to answer in 20 seconds, do you think cancer is treatable, yes or no? I will go, no. It's not really what I think, is a part of what I think. I think that now it's not treatable because of all the things we discussed, but the answer I will give is to polarize my opinion, because I have 42 seconds and I have to reduce my opinion in 2 lines. I mean, with Pierre, we discuss always this because I'm very hypersimplifying. He talks a lot on this. And I think he's right, because if you can have, because opinions are always with nuance, and it takes a bit of time to take it out. So I think it's very bad, because also with young researcher, you tend to polarize the opinion then very much.
My opinion on that is that it's a responsibility that we have to communicate, and it's also a responsibility to say to people what we can achieve and what we cannot achieve. And this is why, you know, I mentioned this at the beginning, but the funding agencies, specifically in Belgium, for example, the Foundation Against Cancer, they really try to do this by, how do they do this? They bring the donator, the people that are part of the, you know, funding of the foundation, to the labs. And it's not, you know, really much to, I mean, of course people want to see, you know, what they put their money in, but it's also to understand that if you give, for example, I receive 800,000 euros to develop my scientific program, sponsored by a Foundation Against Cancer. It's a huge amount of money. If you think about me, sometimes I donate to Foundation Against Cancer, but you know, what I give is ridiculous. So you would say, okay, this is impossible. This is, what can you do with such an amount of money, right? And I think it's important for us to show this, to show what we can achieve and what we cannot achieve. It's true that journalists, they push you to, you know, to be oversimplistic, and it's sometimes a risk to say, okay, well, a new treatment against melanoma. This is what, you know, the title of the newspaper on our and then you have the television coming to the lab and say, okay, tell us about your new treatment that you have. No, I mean, but people need this. My mom needs this. She tells me, Pierre, this is great. Tell me about your discovery, and then I have to tell her, look, yeah, I mean, this is part of, you know, the knowledge. So I think we are responsible for this still, and we have to be careful. And that's the same with the young guys. When they come up to us to say, okay, I have this data, what does it mean, right? So we say, okay, this means this. But the important question is also what it doesn't mean. You know, what is the data not telling you? You know, what is it that you cannot say? Because, okay, it offers perspective, but this is important that this says that, I mean, you don't know if this is going to work or not. And what do you need to do to actually address that question? I think this is important to communicate. Having said that, I think it's very important to communicate. Also, I mean, for cancer research, it's obvious that this is really stimulating, the funding of the research. So, here in Belgium, Televi or Foundation Cancer, they do this, and I think that this is their job to comprehensively put the problem on the table and not to lie to people, to say we're going to give you 30,000 for your PhD and then you're going to cure cancer. That's not going to happen. I mean, I think it's an important nuance to bring here.
For example, in this Belgium association, they do this very well.
Yeah.
Fondation contre le cancer and
It's
Televi,
education. You educate the public
it's
to
really,
do it.
it's like Teleton in Italy. It's really.
Having said that, I mean, we spoke a lot with investors, so investors funds, and that's the same thing. So when you speak to them, they ask you, questions. So it looks like what you have is going to do this and this, and they, like general public, you lack nuance. And our scientists were probably excessive in nuance. We do too many nuance, and this is sometimes not seen as a good thing for, companies or business development. But still, I think it's important. I mean, you cannot just say, if you put the money on my project here, I'm going to give you a drug in 3 years. That's not going to happen. So don't, and you still need to invest on the knowledge. So it's not only on the product. You also need to understand what the product is going to be good at, and also, importantly, not good at for patients. For example, it's very important to know what are the patients that are going to be responding to your drug. But it's equally important to know what are the patients that will not respond to your drug. So you don't want to give the drug to someone that will not respond to the drug. So that has the same importance as to know if the patient is going to respond. If I have a medicine today, if you are a responder or not a responder, I'm going to give you the drug and not to her. But if I don't know, I will give the drug to everyone here and then wait and see who is responding. That's a waste of money and so a big risk for public health. So nuance.
Okay. Well, thank you very much, and I think that we can now thank you.
Thank you.
Thank
you.